Good afternoon, or good morning, as the case may be. Hey guys, so this is going to be the first of your lessons this week. There will be another one posted soon. As per the video that I did on the syllabus for digital learning, you will be expected to do these lessons on Google Classroom on the date that they are indicated. So on the lesson, it'll say that this lesson is for 317. Now, it doesn't mean that you absolutely have to do it on 317, which is today. It just means that it is recommended that you watch the lecture on 317, and you will have until tomorrow, 318, at midnight to be able to complete the associated assignments with the lecture. So it's like lecture one day and assignments the next. This is a way to help you to sort of balance the amount of time that you're spending on this and to make sure that you are able to keep up with the work. This week we're only going to do 2 lessons, but next week we should have 3. So I will continue to keep you informed of the situation, but until then we're going to treat this like a normal class and just dive right in. Today we're going to be talking about the comparison of photosynthesis and respiration. So after this, hopefully you'll be able to breathe a little easier after we shed some light on this issue. All puns are severely intended. Okay, so let's go ahead and dive right in. The first thing we're going to talk about is a comparison of the location. We know that the cell respiration occurs in the mitochondria. In and around it, because remember that both glycolysis and fermentation happen outside of the mitochondria in the cytoplasm of the cell. Remember that this is significant because no oxygen is required for glycolysis or for fermentation, so those processes are just to basically regenerate NAD plus without needing any oxygen or the Krebs cycle or the electron transport chain. So those processes occur outside and keep the lights on, basically, just produce enough ATP for us to keep going. The Krebs cycle will occur in the innermost space of the mitochondria, known as the matrix. That is the space that is inside of both of the mitochondria's outer membranes. And then there's the electron transport chain, which helps to ferry hydrogen ions and electrons across the membrane between the matrix, which again is the innermost space, and the intermembrane space, which is the kind of sandwiched little space in between the mitochondria's 2 outer membranes. So this is very different than photosynthesis. Obviously, they happen in 2 different organelles. This is the mitochondria. Photosynthesis occurs in the chloroplast. So the chloroplast is a different specialized organelle within photosynthetic organisms, usually plants, but it doesn't have to be. Remember that plants also include mitochondria as well, so don't think that cell respiration or mitochondria only happens in animals. It happens in plants as well. I mean, you have to be able to not only make your food, but eat it too. So photosynthesis in chloroplasts is going to occur mostly across the thylakoid membrane with the light-dependent reactions. The thylakoids, remember, are the little stacks of green pancakes that are inside of the chloroplast. They're floating in the jelly-like substance called the stroma, which is where the light-independent reactions occur. So remember, light-dependent reactions are going to be charging up all those molecular batteries to be able to power the light-independent reactions that are going to make our sugar. So there are some structural similarities of note between the mitochondria and the chloroplast that we definitely need to pay attention to. The first is the sheer number of membranes. Now that number of membranes usually comes from, or it's cited to have come from, the endosymbiotic theory, which is basically that originally mitochondria and chloroplasts were their own free-living organisms. They were like bacteria that were kind of just, hanging out and living outside in the outside world. And then they were engulfed via phagocytosis, which is a form of receptor-mediated endocytosis. If you're looking at me or at the screen right now being like, could you speak English? I'm just trying to say that they got eaten by something a long time ago. And when they got eaten, of course, whenever our cells eat something, they put a membrane around it. But the bacteria, or in this case, the mitochondria and the chloroplast, already had an outer membrane. So it just kind of added another membrane on top of their own. And then for whatever reason, instead of digesting them, the organism kind of kept them inside and just used the resources that they produced and gave them food so they could keep living inside of them. And eventually, the mitochondria and chloroplast just became a part of the cells. Some of their DNA was sent to the nucleus and was co-opted so the nucleus can control production and maintenance of them, and then they were just a part of the cell. What's really interesting to think about, and what we talked about when we talked about, what is life and all that stuff, is that the mitochondria and the chloroplast, they essentially evolved to become non-living organelles within a larger system. It was more evolutionarily advantageous for them to basically evolve to be dead than it was to be alive. That's really interesting. If you really think about it, that really means that it blurs the line between living and non-living. You could evolve to become non-living and you'd be more successful. That's fascinating. And it really makes me wonder about the future of humanity and artificial intelligence and technology and all that stuff. But I won't get into that. Let's go back to what we were talking about. So that double outer membrane, whether it came from endosymbiosis or not, is great for maximizing surface area. So remember we talked about surface area is going to be that total surface available to us on the cell membrane, and that can allow for more things like transport molecules and embedded proteins and all that stuff. It can also provide more of a space for molecular interactions between enzymes and substrates to occur. So this is obviously very important. We want a lot of reactions to be happening on the surface of our mitochondria and chloroplast so that we can make sure that a lot of photosynthesis and respiration is happening. If you look inside of the thylakoid and in the intermembrane space of the mitochondria, you're going to see that there is a very small internal space. So I'm actually going to go ahead and I'm going to show this to us in a diagram version. So if we go over here and look at the mitochondria here, this is the outer membrane, this part right here, and then this part, this little curvy structure on the inside, we can see it's like a whole nother container inside of a pill. This part is the inner membrane. So the little space here between the outer membrane and the inner membrane, this space right here, this is the inner membrane space. And that's where all the hydrogens are going to pile up. And notice that the inner membrane space is quite a bit smaller than the total maximum volume of the outer membrane or even of the inner one. It's kind of this little weird in-between space. And most of the inner membrane space that's going to be used by cell respiration is going to happen in these little folds. And these folds increase surface area, but they also make a very small space, making it easy for a hydrogen ion concentration gradient, which is what we need to flow through ATP synthase to build up very quickly, which is good because that just increases the speed and efficiency of the reaction. Here's a chloroplast for comparison, right? We have the outer membrane of the chloroplast, and here's the inner membrane. It does have an inner membrane space, but it's not utilized in chloroplast like it is in mitochondria. Instead, we have these little flat pancakes or cookies or whatever you want to call them. They look kind of like moon pies to me now that I think about it. And they're thylakoids, right? And we call a stack of them a granum, and the thylakoids are all connected, and they have all these folds and stuff, and that allows them to increase their surface area enough to run a lot of these reactions. Going back just one thing, sorry, this is the matrix of the mitochondria, right? Inner membrane space and outer membrane. And this over here, this would be known as the lumen. Well, the lumen is actually on the inside of these little cookies, so it's the cream filling of the moon pies. And then the stuff that all these thylakoids is floating in is called the stroma, right? Okay, so those are the structural similarities. There's actually, there's just one more I want to talk about here. And besides the small internal spaces that promote rapid hydrogen ion gradient for chemiosmosis, which, by the way, that just means literally the buildup of hydrogen ions or other molecules so that they can flow like water through another molecule, like ATP synthase. Remember that osmosis is just a passive flow of water from high concentration to low. So in this case, it's the same thing. It's just not water, it's chemicals. So anyway, sorry, got myself a little bit distracted there. The last similarity is that they both, the mitochondria and the chloroplast, both utilize electron transport chains. I want you to notice where they are. So the electron transport chain in the mitochondria is happening in the inner membrane space in the matrix, so that membrane between the matrix and the inner membrane space. And it's also in the chloroplast, it is happening in the membrane of the thylakoid. Both electron transport chains are going to facilitate the pumping of hydrogen ions into whatever space to promote chemiosmosis and the phosphorylation, or if you will, addition of a phosphate group onto an ADP to produce ATP. So they both produce ATP. They are just located in different membranes and different organelles. Okay, so let's talk about the purpose. So we know the purpose of photosynthesis is to take light energy and to make it into the energy of sugar. We said that sugar was like an unrefined fuel that couldn't be used immediately by the body, but it was an important intermediate between the super unrefined stuff and the fuel your body actually uses. So we know that photosynthesis is split into two different main reactions. There's the light-dependent reaction, which is only called the light-dependent reaction, and that uses the light to split water into hydrogen ions and electrons and oxygen. The oxygen goes away, and the hydrogen ions are used in the osmosis to phosphorylate ATP or ADP into ATP. And the electrons are using electron transport chain to power more pumping of hydrogen ions into the lumen of the thylakoid, but also in order to pass those excited electrons on to NADP plus, so that NADP plus can be reduced into NADPH. So remember that phosphorylation, again, is just the addition of a phosphate group onto another molecule. In this case, the phosphorylation of ADP, which is adenosine diphosphate, turns it into ATP, adenosine triphosphate. So we add on that extra phosphate group, which increases the molecule's instability and therefore its reactivity and ability to power molecular reactions. The second reaction of photosynthesis we know is the light-independent reaction, happens in the stroma. It's also called the Calvin cycle, or sometimes the dark reactions, although that is mostly a misnomer. So I prefer for you to use either the Calvin cycle or the light-independent reactions. And that uses those molecular batteries that we charged up in the light-dependent reactions, the NADPH and ATP, to power the fixation and reduction of CO2 and all those other molecules that we talked about that we don't need to know too much detail about in the Calvin cycle into glucose and also RUBP. Because remember, RUBP, ribulose bisphosphate, is the molecule we need to reset the cycle and to go again and again. Remember, it is different than ribulose, what is wrong with me? Rubisco. It is different than Rubisco, which is the enzyme that actually catalyzes those reactions. So anyway, through photosynthesis, we end up with glucose, and we regenerate our NADP plus and ADP so that more molecules from the light-dependent reaction can come back and keep powering that cycle. Okay, so aerobic respiration, which I'm going to make sure that I distinguish from aerobic or anaerobic respiration, anaerobic respiration, which is the respiration in the absence of oxygen. We're going to mostly talk here about aerobic respiration because remember that anaerobic respiration or fermentation is basically just glycolysis being used to make enough ATP to keep the lights on and to try to regenerate NAD plus. But in this case, I want to focus more on aerobic respiration because that is the focus of most of IB stuff. So remember that aerobic respiration's function is to take all of the glucose and sugars and carbohydrates and everything produced by photosynthesis and to convert them into the refined fuel of ATP, so the stuff we actually use in our daily lives. So the first process, glycolysis, is just going to oxidize or remove electrons from glucose, and it's going to use those electrons to reduce NAD plus into NADH. So there's our first electron carrier getting its electron. It's also going to take some phosphate groups and phosphorylate ADP into ATP. So we create NADH and ATP, the same molecular batteries we created in the light-dependent reaction. We are then going to take the pyruvate that was created by the splitting of glycolysis, and we're going to take that pyruvate and we're going to move it into the mitochondria, to the matrix of the mitochondria, that innermost space where the Krebs cycle or citric acid cycle can occur. You will sometimes see it called the citric acid cycle. So acetyl-CoA is created when we split some carbons off that pyruvate. The carbons join on oxygen and become CO2. And the reduction of acetyl-CoA, so the addition of more electrons, and also the oxidation will power the reduction of NAD and FAD to NADH and FADH2. So we're basically popping off electrons and putting them onto NAD and FAD so that they can carry those to the next phase, the electron transport chain. It will also, again, phosphorylate ADP to ATP. That CO2 I mentioned before was released from the extra carbons we popped off of the pyruvates to make them into acetyl-CoA. Remember that acetyl-CoA is the necessary molecule in this reaction to get this cycle started again. It's much like RUBP. and the Calvin cycle in photosynthesis. Okay, so finally, we get all that NADH and FADH2. It's going to carry those electrons from the Krebs cycle to the electron transport chain. And again, we're going to use the energy from those electrons to go ahead and pump hydrogen ions into the intermembrane space, that very small, thin sliver of space, so that a chemiosmotic gradient can be established. The hydrogens can flow through ATP synthase, and we get ADP phosphorylated into ATP. Because this process uses oxygen at the very end to gather up those hydrogens that flowed through the intermembrane space, we call this oxidative phosphorylation. Whereas because we used water being split by light in photosynthesis to power this reaction, we would call that photophosphorylation. So just to know that phosphorylation is the same, it just means attaching a phosphate group onto ADP to make it ATP. But in this case, in aerobic respiration, we're using oxygen. That's where the aerobic comes from. So oxidative phosphorylation. And in photosynthesis, it is photophosphorylation. Okay, so what are the functional similarities of these two processes? So they both share electron acceptors. So even though NAD and NADP are two different things, they are both electron acceptors that do a similar job of taking the electron and transporting it to the electron transport chain. RUBP is the final electron acceptor in photosynthesis. You could argue glucose is as well if it's produced by the Calvin cycle. And in aerobic respiration, that final electron acceptor is O2 or oxygen. So that's the most important electron acceptor there, and that's why we call it oxidative phosphorylation. So another example of a similarity is a chemiosmotic gradient. So both of them use that hydrogen ion gradient to go ahead and flow through ATP synthase to phosphorylate ADP to ATP. That phosphorylation, again, is similar between the two. That's what they both do. And then they both have redox reactions. So redox is just a way of referring to both oxidation and reduction in a shorter way without having to say oxidation and reduction every single time. They both have these reactions, which are just popping electrons off and putting them someplace else, usually for an electron carrier like NAD or FAD, so that those can be carried to the electron transport chain and power the pumping of hydrogens to create a chemiosmotic gradient to phosphorylate ADP. Good God. Anyway, they also have the same products and reactions, just in reverse order. One process flows into the next. They are part of a big cycle, part of the carbon cycle. Okay, so now go ahead and you can pause the video if you want to, or I guess you could do this afterwards. So be a good time to go ahead and take a little break from my voice and check out the Google form and do a little bit of practice to make sure that you understand the differences and similarities between CO2 and respiration. Okay, so I'm going to assume that you did your practice and we can move on. Let's talk about cyclic and non-cyclic photophosphorylation. And you're like, what? Okay, photophosphorylation shouldn't be a problem anymore. We know that breaking it down, it just means that we use light in order to phosphorylate or add a new phosphate group onto ADP to make ATP. That's all it means. But what the heck do we mean by cyclic and non-cyclic? Okay, so we did an experiment, and I know the experiment kept being delayed because it was taking a long time. But we know that when carbon resources are scarce, the photosynthetic rate tends to slow down. And we talked about this before. We said that that was because if we weren't using NADPH to power the fixation and reduction of carbon and RUBP in the Calvin cycle, then we wouldn't regenerate any more NADP+. We'd run out of it. And if we ran out of that, what in the heck was supposed to accept the electron at the end of the electron transport chain in the light-dependent reaction? Well, we're kind of stuck there, aren't we? And therefore, we're not going to be able to get our electron transport chain off the ground, and photosynthesis will grind to a halt. We know this can all happen if carbon is not accessible, right? But how in the world does photosynthesis counteract this? We talked about what would happen if we ran out of oxygen in respiration, right? We said that leptin would be produced, the hormone that would reduce your appetite, and then you would just, not be as hungry and you wouldn't respire as much. But photosynthesis can't quite do that. So how does it handle this situation? Well, Photosynthesis normally runs non-cyclic photophosphorylation, and if you're panicking at this point, it's just what normally happens in the membrane, so don't. You already know this. We know that the electron is split off of the hydrogens, goes up into the photosystem. It's excited by the photosystems 1 and 2, travels along the electron transport chain, helps to pump some hydrogen ions into the lumen of the thylakoid, and then it's attached to NADP plus at the end to make NADPH, right? We already know this. This is the normal thing that happens. And just to assure you that you know this, here is a diagram showing the same thing, right? So we have our hydrogen ions in here, H2O is split, the electrons go up into the PS2. The PS2 gets excited by light, it fires the electron down through these intermediate molecules, it gets to PS1, woo, excited again, boosh, fires it down to reductase, where we get NADPH. So you already know all this, and all the hydrogen ions get produced, and they flow through, and they make ATP. Cool. So that's non-cyclic, and we say it's non-cyclic because the electrons go from PS2 all the way through PS1 into the NADPH, and then they just keep on going. They don't kind of come back, if you think about it. But what happens in cyclic? Okay, so in cyclic photophosphorylation, we end up getting the photosystem to skipped. Okay, so what do I mean by that? So let's go back to this diagram really quick. When we look at photosystem 2 and photosystem 1, if we ran both of them at the same time, so an electron gets excited over here, it starts going over here, and another electron comes up here, we could hypothetically have 2 electrons charging at the same time, one in PS2 and one in PS1. PS1 fires its electron on, PS2 fires its one on to PS1, and they keep replenishing each other. If that's happening, we're creating NADPH at a super fast rate, right? They just keep on flowing, keep on flowing, keep on flowing. If we're creating a crap ton of NADPH really, really fast, but we have no carbon, we're going to end up using all of our NADP plus way too fast, right? And we're going to basically have a burst of activity and then nothing. We do not want nothing to happen, right? Because we need a slow release of this stuff so we can keep our cells alive if we're a plant, or keep our cells producing sugar, glucose, which is living. So how does it counteract this? It shuts down PS2 and only allows PS1 to function. Okay, how does this help? Well, if PS1 is the only one allowed to function, then we slow down the amount of NADPH that's being used. It makes it so that NADPH can be used in the Calvin cycle with what little carbon we have left, and enough of it can come back and catch the electrons in time so that it doesn't gum up the whole system. So essentially, we just slow down production by shutting off one of the big generators and leaving the other one going. So cyclic photophosphorylation is how plants deal with this. Remember that leptin production in respiration is how they dealt with that. And I said pepsin, excuse me, I meant to say leptin. Leptin production decreases appetite in respiration and slows it down. And this, in plants, we use cyclic photophosphorylation to just run PS1 and to slow down the process. Okay, so what's another difference? We know that the power and the energy for cell respiration does not come from the sun. It actually comes from ATP that we already have in our bodies and all the chemical energy we have stored up. But photosynthesis has to get its energy from somewhere. It gets it from the sun, we know. And photosynthesis gets it from the sun and channels it using molecules known as pigments. All the pigments in photosynthesis are crowded within PS1 and PS2. Those are our photosystems, is what they're called. And within the photosystems, the pigments are going to absorb different wavelengths of light. Now, most of the pigments, most of the heavy-hitting pigments in plants are chlorophyll A and B. They're going to be the ones that absorb most of the light on the red and the blue spectrum, and they're going to reflect what they don't absorb, which is the green. However, they're not alone, because if you think about it, different times of year, different amounts of light are available, different wavelengths of light, and so they have accessory pigments there to help. Accessory pigments are just other pigments that are hanging around with the chlorophyll, and they can absorb different wavelengths of light, like yellow and green and all that stuff, or they can absorb more red and blue. It doesn't really matter. They're just there to help out the chlorophyll molecules to absorb as much light as possible. But what's really kind of interesting about them is that they are also the reason why trees change colors. So, during the fall, the trees change colors because the light wavelengths that are reaching the plant and the amount of light is not enough to sustain chlorophyll A and B. They can also denature when it's cold, so they denature. And the remnants in the leaves are like carotenoids and xanthophylls, and these accessory pigments are able to absorb green light very well and yellow light and all that, green light and some blue light, but they don't do a good job of absorbing yellows or reds, and so they reflect back yellow and red, and that's why we get that beautiful color change in the fall. But anyway, so pigments are how plants are able to absorb all the light, and they use that energy from the sun to catalyze photolysis, so they split water, and then start that whole chemiosmotic gradient we've been talking about. So pigments in plants, the respiration doesn't really have a similar molecule like a pigment, because they get their energy from chemical energy already in the body. Okay, so here we can see, this is just a visual diagram of the light coming in. It goes through xanthophylls and carotenoids and chlorophyll B, and then finally chlorophyll A. And we can see that along the way, the wavelengths of light are being reduced and reduced. That's because they're being absorbed by xanthophylls and carotenoids. And only the ones that are useful to chlorophyll B and A actually reach them. And then over on the left, we can see the light comes in, and they bounce around the photosystems in things called antenna pigments. These are just pigments that are all next to each other in these little cells, and the light gets bounced around, excited, until it reaches the center of the photosystem called the reaction center, and that's where the electron is. And it powers up the electron and then fires it to the next molecule in the chain, which is the acceptor molecule. And there we go. We start our electron transport chain. So just a cool diagram to help with this. Okay. So we're almost to the end, don't worry. We've talked about what happens when there isn't enough carbon and all that stuff, but what about when there's not enough water? And you're like, what the heck does water have to do with carbon? Okay, so I want you guys to think back to the leaf diagram we looked at. If we look back at this leaf diagram here, we can see that this is the leaf, the cross-section of the leaf. Here are our waxy cuticle and epidermis and palisade mesophyll and all that stuff. And down at the bottom, we said that these little holes on the bottom, these are called stomata. These little holes on the bottom is where gas exchange occurs. And we said that this is how the plants are able to take in CO2 from the outside air, right? So they take in the CO2. But when they do that, this airspace, it isn't just air, actually. It's also full of H2O. So water vapor likes to hang out in this little space, and the water vapor is coming out of this vascular bundle, which contains xylem, which is basically the veins in the plant that carry water, and the xylem is going to help to inject that water vapor into the air in the leaf. So when the CO2 goes in, when the stomata open, the CO2 goes in, not just oxygen, but all that water in the oxygen, that water vapor is going to exit the plant. And you might think, what's a little water vapor going to do to the plant? Not much, right? But actually, that water vapor is a lot of that plant's water. In fact, it's most of that plant's water. So that plant is losing water hand over fist as it opens up its stomata to take in the necessary carbon it needs to make sugar. This is a huge problem, right? Because water and sugar are both vital to the survival of the plant. So the plant has to decide, is it going to take in CO2 at the risk of losing water, or is it going to wait for a little while at the risk of not having enough CO2 to make enough sugar? So this is a constant struggle for the plant, and it has to decide whether or not it has enough water to be able to spare to take in CO2. And you guys are going to kind of look at some ways in which plants are able to deal with this when you do your independent research. But for now, there is another problem. So besides the water loss, and you guys will look into that more in the research, as the plant opens its stomata in order to take in CO2 or not, it runs into the problem of photorespiration. And you're like, oh my God, is photosynthesis and respiration combined? Not entirely, not exactly. So photorespiration refers to the fact that rubisco, which is that enzyme that usually catalyzes a reaction to attach the carbon from CO2 onto RUBP to start that whole Calvin cycle thing. Rubisco can also attach oxygen, or O2, onto RUBP. Now, this sucks because when that happens, it produces an intermediate that makes it impossible for us to make glucose. We can regenerate RUBP, but it doesn't make glucose, which is useless to us, right? Because the whole point is to make glucose. So photorespiration severely reduces the efficiency of the photosynthetic process, and it uses up RUBP that could otherwise be used to make glucose. Now, well, why would this happen? Okay, so this happens because there's not enough CO2 close enough to Rubisco for it to actually add it onto REBP. So when those times when the plants are sitting tight and they can't afford to lose a lot of water, there is more oxygen surrounding their palisade mesophyll cells and their chlorophyll and all that good stuff than there is CO2. Because of that, Rubisco is going to grab the first thing it sees, which is probably going to be oxygen. And when it does that, it's going to start the process of photorespiration and make that useless intermediate. So glucose production is going to become severely limited. This is a terrible thing. The plant has got to figure out some kind of way to counteract this process so that it can survive even when there is not a lot of CO2 available to be pulled in through the leaves. So this is a little quick diagram to sort of explain that a little bit. So on the left we have photorespiration. Just like on the right, we start with REBP, but Rubisco uses oxygen instead of CO2. When it does so, this is going to produce useless intermediates that still produce CO2, funnily enough, but it off-gases it and then just goes back to regenerate REBP. Now it does use ATP and NADPH, so maybe this would help us if we were having problems with our electron transport chain, but it would not be good for the plant because it would not produce sugar or glucose, which is the goal. Okay, so now you guys are going to do a little bit of independent research to see how plants overcome some of the issues that we've just talked about. How did they overcome the whole thing about not being able to open their stomata to take in CO2 for fear of losing water on hot, dry days? What do they do if they're desert plants? All that good stuff is going to be found on that Google document. And that is the end of this lesson. So your assignments for this lesson are to complete the Google form practice and the Google Doc independent research. Turn those in to me by tomorrow, which is the 18th of March at midnight, and you guys should be good to go. Look for the second lesson. It should be posted soon. I hope you guys are staying safe and well, and I look forward to seeing you again in person the first week of April. Take care.